Bismillahirrahmanirrahim. Today we are going to talk about myelin sheath. What is myelin sheath? What is the formation of myelin sheath? Okay. What is myelin sheath? Myelin sheath is a thick lipoprotein sheath, lipoprotein thick layer around the axon of the myelinated neuron. Okay. As we know that every axon is supported by a supporting cell, and the central nervous system, that supporting cell is called oligodendrocyte cell. But in the peripheral nervous system, axon is supported by the Schwann cell. These supporting cell produce a supportive and insulating layer, thick lipoprotein layer around the myelinated axon, which is called myelin sheath. Okay. Suppose this is a neuron. This is the axon of the neuron. This axon is the Schwann cell. These are the Schwann cell. These Schwann cell. Suppose this is the neuron of the peripheral nervous system. So the axon of this neuron is supported by the Schwann cell. As we know that every axon, this is the simple axon. This is the axon. Every axon, myelinated axon or non-myelinated axon, is supported by a supporting cell. It's called Schwann cell or oligodendrocyte cell. But this is the axon of the peripheral nervous system. Here present a Schwann cell, and this is the nucleus of the Schwann cell. This is the non-myelinated axon, but this is the myelinated axon. In non-myelinated axon, only the Schwann cell partially cover, only the cytoplasm of the Schwann cell partially cover the axon of the neuron. This is the non-myelinated axon. Okay. But in the myelinated axon, the Schwann cell will make mini wrap around the axon. They make a thick lipoprotein layer. That lipoprotein layer is called myelin sheath. Schwann cell is the cell with a single cytoplasm and a single nucleus. Okay. But the myelin sheath is a thick layer of the lipoprotein, which is made from the mini concentric layer of the cytoplasm of the Schwann cell. Let's see here. When I zoom this area, this is the axon. This is the one concentric layer, two concentric layer, 3, 4, 5. One Schwann cell make concentric layer about 40 to 50 layer around as the myelinated axon. When this Schwann cell make mini circle around the axon of the neuron, they make a thick layer. This is the longitudinal section. This is layer number 1, 2, 3, 4, 5. It make mini layer, so it make a huge mass. First stage, this is the cut section. This is the axon. Suppose the concentric layer start from this point. So when this is the axon, this is the cell membrane of the axon, which is called red color, which is called exoplasm. This is the exolemma. What is exolemma? Exolemma is the cytoplasm of the axon. This is the cut section. This is the axon. While suppose this is axon, this is the, as you see here, this is the cut section. In the middle, you see the carbon material is the axon, and around the circle is the wooden material is the myelin sheath. Suppose The myelinogenesis starts from this point. So the nucleus of the myelin sheath, the nucleus of the Schwann cell, is now present here. When they make circle around the axon, the nucleus is pushed forward and outward. The nucleus is pushed forward and outward. Finally, the nucleus reached up to the top position. When the Schwann cell cytoplasm, suppose this is the axon, when the Schwann cell cytoplasm make mini trip around the axon. At first stage, these strips are loosely arranged, but with the passage of the time, these strips from this point up to this point, these layer strongly fuse with each other and make a condensed layer, which is called myelin sheath. These are the loosely layer of the myelin sheath, but here this is a strong sheath of the strong layer of the myelin sheath. Here is the axon. But in the condensed state, now there the yellow color is the this is the nucleus of the myelin, and this is the nucleus of the Schwann cell. But in the condensed state, this is the mature. This is the mature myelinated axon. This is the non-mature, because these are loosely arranged. Now at the mature axon, the Schwann cell is now present at the top. So the Schwann cell have its own separated cytoplasm, which is separated from the condensed mass. This is a condensed mass. So what is Schwann cell? Schwann cell is the cell in the peripheral nervous system. This is a supporting cell. Suppose this is the supporting cell. This is the cytoplasm of the supporting cell. This is the cytoplasm of the Schwann cell. This is the nucleus of the Schwann cell nucleus. Okay. Schwann cell is the supporting, protective, insulating cell in the peripheral nervous system, which make myelin sheath and the myelinated axon in the myelinated neuron. Okay. The myelin sheath is made by the Schwann cell in the peripheral nervous system, but in the central nervous system, myelin sheath is made by the oligodendrocyte cell. Okay, the chemistry of the myelin sheath of the central nervous system and the chemistry of the myelin sheath of the peripheral nervous system is different. The composition of the myelin sheath is different, but the function is same. What is the function of this myelin sheath? The main function of the myelin sheath is to insulating the axon, just like a wire. The metal part of the wire is insulated by a plastic covering, so the electricity in the wire from the power station is a flow in easy way. Other function of the myelin sheath is to provide quick transport, flow of the impulse in the nervous system. What is the function? The myelin sheath is the disintegrate sheath. It is not a continuous sheath. This is the sheath number 1. Here, as you can see here, this is the nucleus. The uppermost layer, this one is the cytoplasm of the Schwann cell. But this is the condensed. Myelin sheath. This is the condensed myelin sheath. These are the condensed myelin sheath. But the upper layer is the cytoplasm, is the true cytoplasm of the Schwann cell. Okay. As you can see here, the distance, the point between two myelin sheath or two Schwann cell is called node of Raynaud. This is the node of Raynaud. This is the point between two sheath or two Schwann cell. But the distance between one node of Raynaud and another node of Raynaud is called internode. This is the internode. What is the main function? What is the function? Number one, the function is to provide quick impulse. Quick impulse. Suppose when I touch this area, this is the dendrite of the peripheral neuron. The action potential is produced here, and here the action potential is processed in the forward direction. How action potential is produced? Suppose I am say a supposition point. This is the point. When I touch this point, when I stimulated this point, how action potential is produced? When I touch this point, sodium influx start. Sodium go inward. Sodium start inward, and the potassium go outward. So the action potential is start from this point, which run from this point up to and go in forward direction. So the action potential flow, the impulse flow in the forward direction. Okay. How this action potential flow? When the sodium go inward, potassium go outward, the action potential is move in the forward direction. Here the action potential is produced when the sodium come. These are the sodium. Then this sodium start deep. This sodium come to this point and start depolarization. So here the sodium start inward, potassium start outward. This sodium, when sodium go inward, this sodium come to this point and start depolarization. Sodium start inward, potassium start go outward. So and the myelinated neuron, the action potential, the impulse move in the forward direction in the form of jumping. This is the myelinated neuron. But in the myelinated neuron, suppose this is the non-myelinated neuron. This is the non-myelinated neuron. When I touch this area, sodium start move inward, sodium start move inward, potassium start move outward. Sodium start inward, potassium. So it will take more time. But in the myelinated neuron, when this sodium go inward, this sodium, some of this sodium go from this point up to this point. This is the non-permeable. This area, this insulated area is non-permeable to the inflow of the sodium and outflow of the potassium. This is insulating area. Here the ion does not go in outward. See? Okay. So in the myelinated neuron, the some of the sodium is come to this point. It activate this area. When this area activate, the sodium is in this area start move inward, potassium start outward. So the action potential is jump from this point up to this point. So when sodium go inward, some of the sodium come to this point and activate this area, and so on. This so in the myelinated neuron, the impulse move in the forward direction and the jumping manner. So this is the quick impulse. This is mainly occur in the reflex action. This is the reflex arc neuron. Okay. But in the non-myelinated, this is the non-myelinated neuron. The impulse go in the forward direction in a slow way, because there is a continue inward of sodium and continue outward of the potassium. So it will take more time. Okay. This is the function of the myelin sheath. Number one, it give quick impulse, and number two, it give myelination, so the ion does not move outward and inward in the neuron. So the impulse go in the forward direction in a quick manner. Okay, this is the function of the Schwann.